Hello, everyone, and welcome to today's webinar, QC Considerations for Flow Cytometry and Imaging Particles. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit www.beckman.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Michael Collis, Application Specialist at Spherotech Incorporated. For a complete bio on our speaker, please see the presenter component on this screen. Michael, you may now begin your presentation.
Thank you so much, Cassie. I will be sharing my screen momentarily. And thank you and hopefully welcome. Hi. I'd just like to start off by saying thank you so much to Craig and the Beckman Coulter Life Sciences team, to Cassie with LabRoots, and of course to my boss and mentor, Brian Shaw, who's taught me much of what I know. Today we're going to be seeing QC.
Sorry to interrupt you while we're live here. We are not seeing your screen, so if you want to go ahead and end that screen sharing, go ahead and try again.
Oh, pardon me.
Want to make sure everyone gets the information they're here for. There we go. Perfect. All right, I'll pass it back over to you. Thanks, Michael.
Thank you so much. And not to repeat myself, but thank you so much for all being here, attendees, Beckman Coulter Life Sciences and LabRoots, Cassie and Craig for supporting us here, as well as Brian. QC Considerations for Flow Cytometry and Imaging Particles. Again, this is me, the Technical Application Support Representative. On the agenda today, so what we're going to try to, I know a lot of you may be familiar with Flow, some of you may be new to Flow, could be your first time, could be a review. But we're going to go over some basic goals and objectives for QCing and utilizing your flow cytometer. So we'll be looking at alignment and calibration, as well as extrapolation for the quantitative intensity identification of unknown populations using MEF. And we'll get into a lot of these things if we have time at the end, or we probably will touch on background noise and low-end sensitivity, but it's not a direct focus here today. This is a bit of my biography. I'm just a conduit of information for you. There are many giants and rock stars in the field of flow cytometry, and I'd rather highlight them and point you to their literature, such as Joanne Lanigan, Kathy Muirhead, Dagna Shearer, Jolene Bradford, Paul Wallace, Bruce Bagwell, Leonard Herzenberg, Albert and Vera Donnenberg, Paul Robinson, John Martin, and Mark Wilder. I think if you're really looking for great publications, and many more than I could even name here, those are some good places to start. Also, use your resources. I won't touch on it, but I will try to remind you, use your flow manuals, check your own configurations, and there are great courses and conferences to attend. For example, there's the UW, University of Wisconsin-Madison, has an annual flow course. There's the ICCS conference, the International Clinical Cytometry Society, GLIFCA, which is close to us, as you'll see soon, the Great Lakes International Imaging and Flow Cytometry Association. And then CYTO is kind of the big one here, the International Society for the Advancement of Cytometry. Now, in my biography, the one really cool thing that I'm very thankful for is being able to go to the University of Loyola for some research. I didn't at the time know. I was probably like many of you. I was thinking I want to study biochemistry. I might want to do something in genomics or proteomics. I was even thinking of crystallography, and I ended up working with some Central American cichlids, more famously the African cichlids, and their sequencing of their photoreceptive proteins. It's very cool how some of the best technology mimics biology. So when we think of a lot of our channels and looking to a lot of these flow configurations, dichroic mirrors, bandpass filters, in a lot of ways, them in concert with the photomultiplier tubes, we'll get to all of these, are very similar to your eyes, your cones, as we call opsins, right? So they're going to cover, like we're trichromates. We have a short wave sensitive, a mid wave sensitive, and a long wave sensitive cone, as well as a rhodopsin. And we're going to see these kind of re-represented in flow cytometry by looking at front and side scatter, kind of like your rhodopsin. And then your fluorescent channels are going to be kind of like our cones. And a lot of your instruments, if not currently, may have originally only been off of maybe 2 lasers and only had 3 filters, which is very similar to us. These wonderful fish that I was able to sequence from have a concert more similar to like a 13-color Beckman-Coulter Cytoflex or many of the other robust instruments that we'll find in the field. So I just wanted to point that out. What's Spherotech? Maybe this is the first time you're hearing of Spherotech. We're a company that's found on the Great Lakes, just north of Chicago. And we were established in 1992 with our founder, Jeff Wang, a brilliant chemist, a polymer chemist, as well as organic chemist in the field. Some of the fluorescent dyes that we utilize were actually synthesized by him and are available inside of our particles, which is really, really cool. He's the one who originally polymerized the polystyrene that we now use today. And we're still a family-owned, fairly small organization. As you see here, we have between 36, maybe as high as 40 employees now, and somewhere between 25 and 30 of them are staff scientists. So we do have the ability to be small and agile for custom work, as well as robust enough with our over 150 years of manufacturing quality and research and development experience. We're an ISO 9001 2015 certified organization. And as we say, we're committed to produce, design, and deliver quality products that meet established consumer requirements, defined specifications. Okay, so who do we support? You. We support you, right? Whether it be instrument manufacturers, such as the great Beckman-Coulter, and other in vitro situations. We have hospitals, universities, researchers, private companies. As you see, some of our customers that we're not going to be dealing with today, or some of our clients are in the biophysics field. They're going to be dealing with magnetic particles or ferromagnetic particles and dealing with magnetic moment and all kinds of fun things there. A lot of what we're talking about is going to be on the clinical side, assay development, potentially pharmaceutical companies, and how we can support you to your research goals, typically in the biological, biochemical spheres. So we throw around a lot of words. You'll hear particles, spheres, beads. What are we talking about? Well, we're talking about latex polystyrene, predominantly. We do have a few offerings in polymethacrylate. Divinyl benzene can sometimes be used in order to make those particles a little hardier. We do have some silica. And recently, we've been able to work with some nano gold particles in the field, which is very cool. A very cool opportunity there if that's something that you might be interested in. And just as an example, in the upper right-hand corner here, we have some of our 3-micron polystyrene particles shown on human hair. We never disclose whose hair, but it's an interesting artifact that we have from our own SEM when we were first getting it. So again, why would we use particles? Why are particles important? Why might you care? And it's because they can be used as rulers. They give us insight into this very specific biological range between maybe 100 nanometers, just thereunder, 100 micron. Most cells, biological life, potentially viruses or bacteria, all kinds of fun things. We can antibody coat them. We can use them for all kinds of testing. So they can be used fluorescence, magnetic, functionalized, coated. We have a good concert between carboxyl. We've even included click recently, if somebody's interested in click chemistry, as well as classic opportunities like polyclonal antibodies, protein G, streptavidin. So you can really build out what sort of tools you might need to further your research. Because at the end of the day, we're here to serve you. Often we say, we're the plate and you're the chef. I've mentioned some of these offerings. Today, we're going to be focusing on these highlighted ones, these calibration particles, Rainbow QC kits. Other great tools that you might look into having a flow cytometer might be absolute cell counting particles, so you can accurately determine the size of the population, right, that you're dealing with. Compensation is not something we're going to be touching on today, but is a core competency in flow cytometry. And it focuses more on having exact matches for your floors and being able to compensate. As many of you may know, we could even have a webinar specifically on compensation. Multiplex particle array kits, usually different intensities in long wave sensitive channels, so that you can use things like FITSI or PE as your markers, even things off the violet laser, so that you can develop and multiplex. You can have multiple tests for multiple parameters or multiple assays in single runs, which is really, really helpful, especially if you're dealing with, let's say, bloods or serums or certain kind of identification, purification, but really assay development. And we do offer some slides if you are looking for microscope support. As we mentioned previously, magnetic, functionalized, all this kind of good stuff we could get into if you wanted to in the Q&A section. So, rainbow alignment and calibration particles. Now, these are very cool. We have a concert of dyes that we are internally polymerizing into these polystyrene particles that can then be detected across all of the channels available to you in your flow cytometer. So whether you have a Navios, whether you have a Cytoflex, whether you have a Gallios, a cell sorter, or traditional flow cytometer, you're going to be able to align and calibrate and track the performance of your instrument, hopefully with one, maybe two particles. As I say, like one particle to rule them all, right? We'll deal with alignment with our rainbow, ultra rainbow, and super rainbow. There's only slight differences between them. For example, the ultra rainbow often has an ultraviolet dye internally polymerized as well. Super rainbow might have a slightly more robust, wider range, but we found that the rainbow particles, the original syntheses by our brilliant founder Jeff, still hold in most any flow cytometer, are still the industry standard and are the flagship particle of our organization. You'll often hear in the business, if you haven't heard already, or in the field, 6-peak or more commonly 8-peak rainbow beads. And these can be used for calibration, normalization, MEF, which we'll get into this definition, molecules of equivalent fluorophore, not to be confused with ERF, which we're going to talk about down here, equivalent reference fluorochrome. Okay, so the traditional rainbow calibration particles are offered in two suites typically. One is our traditional, which is what most people are using, the Euroflow Council. We partner with a Euroflow Council in the European Union. It's also in some hospitals in America, South America, different parts of Asia, but you would usually already know that your lab is a member of the European or the Euroflow Council, right? You're going to be using their specific MFI variance ranges. They set some of their own standards. They are fundamentally the exact same kind of particles, but they have specific lots with specific extra parameters that work with their SOPs. It's kind of like using them as a QC outlet or as an SOP source. So if that does sound like something that's interesting to you, or you might want to lean on the validation of a known community, you may want to look into the Euroflow Council to support you. But traditionally, most users are still just using the MEF values set by the standard RCP, such as the 35A. This 30-5 is a 6-peak, and this URCP 38-2K is also a 6-peak. Now this UR is for ultra rainbow, and R is for rainbow. Now if we look a little bit lower, we're seeing this U and S appear with this QP. So we have URQPs versus the URCPs or RCPs. If you are very serious about having quantitative fluorescence, we have NIST certified ERF determined multi-peak and fluorochrome validated particles. They're very specific lots. They're going to be a little bit more expensive, but only slightly. And you'll be able to lean into that certification through NIST, right? They call it ERF instead of MEF because they have actually identified and established which fluorochromes they are using precisely when looking at our particles and assigning them therein. We can look at some, if you are interested, I could show you publications comparing MEF to ERF. Both are highly validated correlative sources. And again, most individuals in the field are still using the MEF values, the molecules of equivalent fluorophore, which are more robust when it comes to the amount of channel offerings, right? But the ERF, although it may only have 4 or 6 components, if you are very tight on your parameters and that is a need for you, we do offer them, which is a wonderful, wonderful offering. And then down here at the bottom, we have this nano calibration. So we do have larger particles, but most often, if you're dealing with a 3, 4 micron, this 30, 38, that's a mean diameter, right? Most often. This RCP-05 is a half micron, 500 nanometer offering. So if you are looking at doing things with EVs, or if you are looking at smaller vesicles inside of serum or whatnot that you want to tag and quantitate, we do have an offering in that range, which is very, very cool. This is an example of these fluorescent dyes. So sometimes people will ask us, hey, you call this yellow, but it shows up in my FTSE channel. It comes off a blue laser and it appears green. Why do you call it yellow? Well, it looks yellow to the naked eye under white light. The same way that our blue dyes, right? You might say, but they come off a red laser. I excite them most typically with a 633 laser, right? They're emitting in the 680, 700. They're showing up in my APC size 7 channels. Why are you calling them blue? Well, they appear blue. And actually, these are the kind of beads when we talk about those multiplex assay particles and we say, do you want a pack of blue beads? PAK, particle array kit, blue beads. This is what we're referring to. We have some examples of our magnetic here as well, but you can kind of see this full concert rainbow of dyes, which I think is very cool. And as you can see with these rainbows or ultra rainbow particles, every dye is internally polymerized into every particle. We're lucky enough to have a robust fluorescent microscope, and we can show that whether you change, if you're looking at the same cluster of particles, and you change the laser excitation or the filter with which you're reading or capturing that emission, the particles continually show up as the same beads, which is great. Okay. This is most generically the inside of what your flow cytometer looks like. This is the anatomy of the wonderful, robust equipment. Some people would say toy. I sometimes say Ferrari. That is Beckman Coulter at your hands, right? You're going to have multiple lasers going through multiple bandpass filters or dichroic mirrors, reflecting through your interrogation of this microfluidics channel. So you're going to have your particles or cells going down this microfluidics channel, being interrogated or excited, and each one of these information will come off of these mirrors. They'll have bandpass filters and mirrors that will then lead to, quite often, photomultiplier tubes, avalanche photodiodes. Sometimes you'll see silica photodiodes. It's becoming more robust and it may change, so most often we just say capture parameters or tools. Sometimes there's compilers. So that's another interesting thing when I say check your manual. If you come to Beckman or you come to Spherotech and you say, I need help in some such a way, it might be good to know your configuration as well as your model. There's sometimes different configurations for the same instrument. If you ever want to upgrade your flow cytometer, you say, hey, I have a great 3 laser instrument. I'd love to add a 4th. How do I do that? How will that affect my configuration? Is that possible? It's great to understand the anatomy of your flow cytometer. These are some very traditional filters. And it's good to know there's sometimes different names for the same filter. So like this 415, 425 filter. Somebody might call this 420-10, right? Because the midline is 420 plus or minus 5. We call it a 420-10. This coming off of a violet laser might be called cascade blue. It might be called pack blue. It might be called like V500, something along these lines. And you might even be able to change the name of your filter channel in your software for your given runs. So it's good to know, are you calling this FTSE? Are you calling it B540? Are you calling it B537? These oftentimes are the same filter or very, very similar, right? We'll get into some names like PE Texas Red, ECD, right? Very, very cool. So this is one example of the anatomy of a flow cytometer. So when you get your particles and you have your flow cytometer, you might ask yourself, what should I do? These come in a dropper bottle. Do I use it straight out of the bottle? Do I dilute them? What am I diluting them with? How do I store my particles, right? Can I dilute them? What are we going to do here? Okay, so they are stable at standard temperature and pressure. You could store them on a bench. Once they're opened, I would recommend putting them in a fridge, 2 to 8 degrees Celsius. It's probably not the most important thing, but what's great about it is they're going to typically come in an opaque bottle. That opaque bottle is probably going to be inside of a foil pouch. So it's going to be protected from light, which is very important. Fluorescent particles do have a chance, if you leave them out in light for hours on end, to dim. So it's good that they're protected. But if you put them inside of a fridge, I know when you close the door, they're protected from light. They're also going to be a little more shelf stable. We do put a tiny bit of sodium azide, about 0.02%, into all of our products, just as antibiologic, antimicrobial, anything like that. Storing them at the fridge could potentially increase that lifetime. We're usually guaranteeing these particles, let's say 3 years after receipt, 1 year after opening. There may be an extended date specifically on the C of A for a specific lot. But if you're still finding them to be in performance over time, it's typically because you've stored them in a very you really want to resuspend the particles before using them. Vortexing, shaking, agitating. You could sonicate them briefly. I wouldn't do it for extended periods of time. But the particles are latex. They're robust. You shouldn't have any issues. Usually we're diluting them. So a drop, as we've measured out of our dropper bottles, is about 25 microliters. But if you put 2 drops into about 1 ml or 2, they're typically coming to you at 1 to the 7th, maybe 1 to the 8th. You're going to have a final concentration about 1 to the 5th, 1 to the 7th. That way, if you run your flow cytometer on very low settings, you're going to be getting between 50 and 100 events per second. And then if you are running them on very, very fast, you could run them at much more robust speeds, but you usually don't need to be running them as aggressively as some people do, although it's great. It can be great for high throughput. You can get a lot more work done in an individual day. But when you're running the rainbow calibration beads or setting up your parameters in each channel before you do those faster runs, this is a fantastic, fantastic dilution. And like I said, these are the 2 flagship, the RCP, Rainbow Calibration Particle, 35A. It's about 3 micron. If you are interested, this 5 at the end is associated with the 5 mils. So 2K is a 2 kit. You actually get the blank and the fluorescent in separate bottles here, along with your 5 fluorescent populations and your 1 blank, making 6. Okay. This is the instrument that I'm lucky enough to be able to run sometimes. Beckman Coulter Cytoflex LX. It's a 13-color instrument. It's very robust. We're very, very grateful to have one in-house. And that's the instrument that I'll be showing you the data for today. I wanted to just give a quick example here. So we're going to look at the alignment particles first, and then the calibration particles. And really what we're doing here is we're making sure that the avalanche photodiodes or photomultiplier tubes are functioning optimally. And you can do this by tracking the MFI, Most people will use median, but you can either set it to mean fluorescence intensity or median fluorescence intensity for single peak particles. Now, I didn't really give a good example of dirty. I didn't want to make my flow cytometer that dirty. But you probably, if you've used not a strong dilution, or if you've put in cells and you're not really sure how much the count is, you could actually have the entire open reading frame pretty well coded in events. But if you look off to this third example, I did want to highlight this concept. This is a doublet. Typically, if you have a population that on a front scatter, side scatter, as we have here, the front scatter is my x-axis, my side scatter is my y-axis. This is run on linear, not log. You'll see this population is just slightly to the right, over and up, kind of in an even distribution, slightly above the main population. So maybe I'm gating these beads and I'm getting an 80% singlets. But I have this second population here, and I know that the standard is supposed to be 85+%, maybe 90+% singlets events. This is likely a doublet. This is what you need to agitate, right? We mentioned agitating, right? Resuspending particles by shaking or agitation. This would be a key sign to me, this doublet, it's possible this is an artifact. If I'm getting over 85+% singlets inside of that P1, but if I'm not, chances are good I should put my instrument on pause, agitate my particles, rerun them, see if I can get that higher singlets percent. And this is an example of what we might see for an alignment particle. All right. Typically, in that first upper left-hand dot plot, I will gate that first population, P1, off of as I said, like an 85+%, 90+% population. Different particles do have different specs. And I usually, as we're running this front scatter, side scatter, on linear, I usually set them at 50-50. You can set them at 100-100. You may already have a QA SOP'd standard for which you're supposed to set these particles. If we look at these other channels below, I'm setting them all to 10 to the 5th, or 10 to the 6th rather. But what's more important is these are on log scale, right? We run our fluorescence channels on log. We run our front and side scatter on linear. And also, if you notice, in that first row, on that linear, underneath 150, you'll see it says like times 10 to the 5th. Or on the left-hand side, it says times 10 to the 5th. You see on the x-axis, it goes 0 to 10 to the 7th, 0, right, to max. You want to make sure that you've expanded your entire dynamic range to the full dynamic range of your instrument. Some instruments will only be 1024. Some instruments will be 10 to the 4th. Some instruments will be 10 to the 7th. You want to make sure that your parameters are full so that you're seeing all of the events possible in your full dynamic range. Does necessarily having a larger number in your dynamic range mean that there's some sort of a gross benefit? Typically not, right? You may get slightly better CVs on an instrument that has only 1024 instead of like 10 to the 4th, because you have that many more bins to kind of measure that confidence variation, that CV. But in general, if a particle is sub 5%, it's sub 5% across the board, right? And I just gave a standard concert of some regular channels. I like to look always at different lasers. So this second row, first one on the left, B525 FITC. That B, I've labeled it for my laser, B blue, 525. Middle one, Y, yellow laser, 585 PE. Third one, red, 660 APC. That's a fantastic place to start. That's where most flow cytometers did start. If you look back into the history of things with those people that I mentioned previously, like John Martin, Mark Wilder, Leonard Herzenberg, these individuals in a lot of ways invented flow cytometry as we know it, and they were only using 1 or 2 interrogation lasers and only had 1 or 3 filters at their disposal. We now have many more, and we're very thankful for that. But you can do just as robust research given these different kinds of parameters. And like I said, if you took like that UW flow course, that University of Wisconsin-Madison annual flow course, you may even be able to kind of build the anatomy of a flow cytometer and kind of really see what it's like to get that pinhole all these various very cool parameters lining up your microfluidics channel to your interrogation lasers. It's very fun. Okay, this is very important. So if we look across the bottom, people will say, how do I know if I'm in a good place? How do I know the variance of my CVs and front and side scatter? How do I know the variance of my MFIs? You need longitudinal data. So a few months ago, I reran these particles because I wanted to have a fresh lot to show for this webinar today. And as we're seeing, we set up the one gate and we ran them continuously from November through December, at least 30 days. I would love to have 60 days as you continue to make your pile of data more robust, right? But at the very least, you would want to have your instrument set up by, let's say, your Beckman Coulter Life Sciences tech. You would want to do maybe a PMT if you already have an instrument in-house and you're not sure exactly where it should be. And you should be running these every single day, 30 to 60 days, recording them, making sure you know what your parameters are, because nobody can set the parameters for your flow cytometer except for you or your SOPs inside of house, your QA, QC team. And as you see, we have this upper line here, probably going to be associated with that side scatter. Side scatter is usually typically higher than front scatter, but you're going to notice that they're only varying within about a 1% or 1.5%. But only after this month could I have said that with confidence, right? And this is the kind of channel data that I'll be gathering, right? If we look at the percent total populations, right, I have that 83%, 89, 90, 87, 90. So I'm seeing if I can always gate that higher percentage that I talked about, 85 to 90% consistently. Then I'm going to look at the CVs and front scatter over time and how much they vary. Usually very tight on these alignment particles. That's the purpose of the alignment particles. This is not typical for what you would see with cells, even fluorescently tagged cells. These are extremely precise particles so that you know if this changes by 0.5%, by a full percent, by 2% in some fluorescence channels, there's something up, right? Now, don't jump to conclusions in your first 2 or 3 days of gathering data, as we'll see, right? In those first 2 or 3 days, you're not always sure what's going to happen, but they should be a normalization. And the numbers may also get better as you continue to run the instruments. And they also tend to have kind of a trend, right? Especially if they're coming off the same laser, especially if they're a similar filter, there should be a similar trend. If you notice, for example, this yellow channel gets really broad, all of a sudden, the CVs are very poor, or the MFI drops. Check another yellow channel. It might be your yellow laser, or it could be your 585 filter. Maybe do you have that 585 filter on another laser? Do you have a blue 585 and a yellow 585? Now, this is really something that your Beckman Coulter Life Sciences tech is going to be able to assert. They may ask you these kinds of questions when you get on the phone with them, but these are the kinds of things that you can do to kind of do some self-diagnosis. What's going on with my instrument? Because it's usually, like I said, the best technology is usually mimicking biology. It's usually not everything's going wrong. It's a chance that it's just one filter. It's a chance that maybe one of your lasers needs to be switched out. There's a chance that maybe somebody ran your flow cytometer until it went dry and they let it go much longer. You always want to make sure that there is a wet tube on your flow cytometer, especially if it's running. If you let it run dry, that could be causing some issues. All right, but you want to be able to troubleshoot this. So, as I said, we can look at various factors. And as I said, we did. So you can also always ask me, hey, what are the MSAS specs for this particle? Now, these are going to be from lot to lot. These won't necessarily be within a specific lot. So like if your FTSE channel is changing in the log channel value and the MFI by 30 or 40% between runs, not normal. Between lots, reasonable, right? There will be a certain amount of time, and maybe I should have covered this earlier. In the good old days, in the bad old days, you had to make your own standards. You were usually culturing your own cell lines with which you would then tag the surface with FTSE, and you would be maintaining those cell lines repeatedly. hopefully so that those cells are going to have the same amount of a specific cell marker, right, on the surface that you can quantitate, and you can say with a certain amount of confidence they should at maturity have a specific range in the MFI, and you would be making your own linearity particles essentially from biological matter. Thank goodness now we have these fiduciary tools. Thank goodness we now have polystyrene particles that we can internally encapsulate with dyes that are extremely regular, where you can dope them repeatedly and you can control the amount to which you're doping them, and you can get them so that you know your lab maybe in Texas is running the exact same things that you're running in California, or your lab in New York is running the same things that you're running in the UK or across the world, right? This is one of the powers of having a specific lot and having polystyrene latex particles that are internally encapsulated with these organic soluble dyes. But there is lot-to-lot variation. On the same beads, between days, no. I want to see if this is 2.5, 2.9, 2.5, 2.3, 2.3, 2.5, 2.3, very regular. Very, very, very regular, right? The MFI values. Now, these are the things that are likely going to change more, because if we look at these parameters, the fluorescent CVs for these channels are going to be under 3. The fluorescent CVs for these channels are going to be under 5. The fluorescent CVs for side scatter and front scatter are under 6, and as we've seen, front scatter is typically lower than side scatter. Those things we guarantee. If you get lot AR01, previously, historically, you had lot AM01, AL01. After a year or two, or a year, maybe 6 months, you completed your bottle, you get a new lot, AR01, AS01. Is there a chance that that new bottle is different from the previous one? Yes. There's something when you're starting to get low, when you're thinking you might get low, ordering that new lot, seeing if they're the same, asking me or one of our other tech support representatives, hey, is this the same master lot? Is this a different master lot? This will come into play a little bit more when we're talking about calibration than with alignment. But these are the kind of questions, these are the kind of answers that you might be seeking. So this is what we mean when we say alignment. We're looking to see, is the MFI, is the mean fluorescence intensity reasonably stable? If I put this peak at 10 to the 6th, or if I put this peak at 10 to the 4th, and this says 90,000 or 50,000, is it going to stay within a specific parameter? It looks like this channel might vary by, this is what, 900,000? It might vary by about 50,000, but it's not really jumping outside of that range. I don't see any 70,000s. I don't see any 100,000s, right? Over time, you're going to have your own confidence parameters for those individual channels. What you see as a normal jump, what you see as abnormal, right? Kind of like if you have kids or family members, siblings, parents, some people's body temperature varies more than others, right? Some people tend to spike more, right, than others. These are things that you'll know what your normal is versus another person's normal. Same with your instrument. Very good. Very good. Making a Levi-Jennings plot, Levi-Jennings plot, keeping this kind of information. It's always good for audits. You always want to be able to defend yourself and say, hey, Michael, hey, Spherotech representative, hey, Beckman-Coulter representative, what are the expectations herein? And where are my SOPs going to be the most important tool? Calibration. So previously, alignment, single peak particle, calibration, multi-peak particle. These are great. They're going to tell me a lot of robust information about my instrument. One of the things that you'll kind of hear from different individuals, different ways you can run these. Again, I start off side scatter, front scatter. I'm going to gate the main population as P1. Some labs will cross, let's say, something off a blue laser, something off a red laser, something off a blue laser, something off a yellow laser, and gate the populations as they increase. Other labs will pick a favorite channel. Typically a favorite channel is FITSI, because FITSI was one of the first instrument channels. It was the one that most of the first publications came out with. They will come along and they will fence gate, as I would say, FITSI, right? Some areas I've seen it. One of the most common places I see it is in hospitals in New York. They have some of the most robust auditing, technical support, QA. It's wonderful. It can also be daunting. They will gate every single peak in every single channel. All of these are normal practices. No lab is better or worse for necessarily doing it. The important thing is, can you track it? And one of the most important things that I do is I like to color code. Now, you don't always have to, but especially if it's one of the first times you're running an unknown, I color code it. Because if I look at a channel and I say, oh, there's only 5 peaks or there's only 4 peaks. For example, this channel, if it was completely uncoded, PE size 7, typically has 1 or 2 of these dim peaks blended. I think it's because of a Stokes shift. I think it's because of the differentiation between the laser line and the filter. What I do know is that in every other channel or many other channels, the resolution's the same. And I also know that across almost any instrument manufacturer, this is an artifact. This is the truth. And you want to know the truth. When we talk about, for example, that 6-peak particle, when I first started flow, I love the 6-peak. I thought the 6-peak is the greatest thing since sliced bread because it always looks clean. Essentially what we've done is we've taken out like that 2nd and 4th peak, so there's higher resolution in every single channel. Now that can be really nice, and if you do need that extra peak for confidence when making your linearity, right? If you have 2 peaks, it always makes a line. 3 peaks, you have slightly more confidence. 5, 6, 7 peaks, you get more and more confidence with each peak, resolved peak that you have in each individual channel. Maybe the 6th peak gives you an extra peak that you won't necessarily get with an 8 peak. However, those giants that I was talking about, those giants that I'm standing on the shoulders of, they know that this is a normal artifact in their instrument. And if they were to have a test in this channel between let's say, a very young, a juvenile, and a mature cell line that they expect has a specific differentiation of maybe only an order of magnitude, less than 2 orders of magnitude, and it's going to be dim, they might want to choose a different reporting protein than PE size 7. Maybe they want to do it in FTSE. Maybe they want to do it in a violet channel. They have very high resolution. If you're not fully aware of the capabilities or resolution that you have in your instrument, you may have false assumptions or hypotheses. So sometimes seeing an artifact is more true than masking one. But again, these tools are extremely robust. Okay, so what I was getting to with this color-coded gating. If I'm running them and let's say I only see 5 peaks in a channel or 6 peaks in a channel, I might think, oh, I remember from the webinar, I remember from readings that I've seen, I remember from publications, some of these peaks might blend. Maybe this is low resolution. Maybe this is the brightest peak. If I've color-coded them in every channel and I see the brightest peak is yellow, I know there's another peak missing. Right? So the first time I run almost anything, I color-code it because that's personally what I like to do. You can look at various other things. Maybe you understand, oh, the dynamic range is 1024. So if I see one of these MFI values that exceeds, like this is 87167534, maybe if this goes beyond what you know is the highest limit for this dynamic range, maybe you're number oriented in that way. I go visual orientation. So I know that every single one of these channels is represented, and I'm putting the 2nd peak. In this case, I'm putting the yellow peak, that 2nd to brightest peak, peak 7 on 10 to the 6th, on 10 to the 6th, on 10 to the 6th, on 10 to the 6th. Being very consistent, always trying to put, which means that every single channel might have a different voltage. I'm adjusting the voltages so that they're showing me a very similar concert. So I can expect one peak beyond that and all of the other peaks to follow suit. So when I run this every single day, I can have that expectation. If I don't want to necessarily get a separate alignment particle, I can use this 7th peak as my alignment particle. I'm treating it like this alignment particle. But this begs the question, why do we need calibration particles? Well, we want to make sure that they're linearized, right? This is a logarithmic scale here. In fact, it's exponential data. We always use the term log because log is how you undo exponential data to make it into a line. We're always trying to fight to make it y equals mx plus b, something that our minds understand. And we can say with confidence, if we put a trend line on top of that, do we have confidence therein, right? And that's what we really want. We want to be able to say something highly correlative about the health of our instrument across the entire dynamic range, from the low end to the high end. Is it consistently measuring 1 log more than every other log, right? Is 10 times always 10 times? So this might be the data that I get. I've grayed out some of the more unimportant things for me. I've put in all the orange lines as my mean channel numbers, and all my blue is my CVs. So if I did want to track them. It's also interesting if we take a moment to look at some trends, because I'm going to mention these when we briefly talk about Q and B values in a little while. 50%, 6, 3, 2, 2, 2, 2, 2, right? These usually are dropping. Very high, much, much lower, sometimes highest, sometimes highest. But there's always a reduction in that CV as the intensity increases. We can see that visually. This CV is much broader than these higher CVs, right? So as we increase in intensity, we're usually decreasing in CV. We're going to take these mean values, and we're going to go to the Spherotech website. We need a template. Underneath this technical tab, there'll be a drop-down. There'll be a templates homepage. If you do want to see, I mentioned some of these before, but like GLIFCA, ICCS, Cyto, we go to these annually. They're important clinical cytometry, and cytometry conferences. We also typically attend AACC, now ADLM. This is our templates homepage. There are some great how-to guides underneath, right? This might be a little overwhelming, but as I said, sometimes lots change. There is a lot of conservation. So from AM01, AL01, AK04, AK. So for multiple bottling lots, they had the same master lot, but there was a change. AK01 is different from AK02. So if you're using a template, it's always good to check. You'll get a technical data sheet with your particles. On the second or third page, you'll see MEF values. Those MEF values should match. Another way that you'll be able to tell is through the linearity, through the actual tool, the calibration tool, the template, your instrument. So these are some Spherotechnical notes. So if we go back here again, this technical tab will drop down. Spherotechnical notes are great how-to guides. Right? We've stripped evident particles. Again, this calibrating, performance tracking, flow cytometry, calibration particles, STN8, STN9, measuring MEF with a flow cytometer, STN14, determining PMT linearity, right? 8, 9, 14. These are gold for what we're doing today. This is what the template typically looks like. These tables, as I've said, some of us have different dynamic ranges. This might be for 10 to the 4. This might be for 1024. You might need to check your manual to be able to determine which one of these is the correct tool for undoing that exponential data, applying the correct log. How do you know? Well, this is a really great way that I like to test, even if I don't know which one I'm supposed to use, because each one of these gets slightly larger. This is a larger log, this is a larger log, this is a larger log. Maybe you need a custom. If I take my data, we call these 256 relative channel numbers. If I put in my numbers, my MFI values from my instrument into these side tables here, and this bottom number is bigger than 256, my data is larger than the logarithmic capacity of this table. I should go to the next one. This one is 5, this one's 9. As you can see, if I put it in there, it's too low. It doesn't actually approach 256 yet. Maybe I've used too low, I've gone too far down in these tables. I've put it into here when I should be putting it into here. This approaches, but does not exceed 256. This is probably healthy. So what we're actually doing is we're taking our MFI data, we're putting it in here. We're getting values. We're taking these values out of here, putting them into the channel number. Right? So this table converts my mean channel number to a 256 relative channel number. This 256 relative channel number goes into this channel number. This is a fixed value. This will change, and we will see this 100, or typically 999, change. So if I gate my particles properly, I've run them, I've gated them in a specific channel FTSE. I'm going to do a comparison between two other channels, but we'll get there in a second. This is a flow chart to show what I'm talking about. I put my channel numbers here. They come out here, this 8892. So if we look, I believe this 3234655. This is that 3234655. This is my FTSE data, right? 3234655, right? 46455. That's where I'm getting these values from my instrument. I'm putting them into this left column. It will give me values out in the right column. I take that right column information, I put it here. These are gold. These we've given to you. These should match your technical data sheet. You will see them inside of the template on different tabs. You will get calculated MEF values that are similar to, they should be similar to, and this will tell you the amount to which it's similar to. You want your residuals to be low. You want your R squared to be nearly 1. This is 9998. This is very good, much better than my grades. You want at least a 997. There might be 1 or 2 channels, like that pesky PE size 7, that might give you a little bit more trouble. But on a FTSE, on a standard violet channel, maybe BV605 is another strange one. But in general, you should be seeing 999, 998. That gives you very, very high confidence that what you're seeing is correlated, just is extrapolating properly. Then we can talk about unknown population determination, this cross calibration. For many of you, this is where we end, right? We're going to put in our data. We're going to try to get this 999. We're going to try to get this extremely low, maybe 0.5, maybe 1, right? We're done. Instrument is calibrated. I can now hand it over to the primary investigator. Instrument is calibrated. I can stamp my QC report. I can say that this instrument is healthy. On this given day, we can show longitudinal data. Many of us stop there. For some of us, we are actually doing unknown population, MEF determination, right? Let's stay here. I know I'm moving around a lot. So let's say I have an unknown population. I have like that juvenile that I was talking about versus that mature cell line. And I don't want to be able to say, this is kind of brighter than that. I want to be able to say with confidence, this has 10, 15, 30 times more, maybe 30,000 times more, right? Binding sites than it did at a juvenile stage. I want to be able to say with confidence, at what point in time is this hormone upregulated? Is this cancer positive? Do we have this specific disease found, right? And to what extent? This is really the power of these particles, of your flow cytometer, is being able to do this unknown population fluorescence determination. So if we look here, we might be able to say, oh, this is a couple of decades maybe brighter than this. What sort of correlation, what sort of confidence do I have on that? Well, if I run my particles and I have 9998 confidence, although I could say, oh, this kind of looks like it's in the same, color, it might be in this same gate P7. I think it might be around, well, let's test it, right? Let's actually take that unknown population, get out that 256 channel number, put it into my cross calibration table. Now, all of these settings have to be the same. We have to hold voltage, right? We need to hold, of course, channel, right? We're going to be holding all the same settings and running them with our calibration particles, then with our unknown population A. Typically, you also have this as a known. You want at least one known before you walk up to your flow cytometer. So maybe you have a fluorimeter, maybe you have one known, one unknown. You're extrapolating, and maybe you only want to know the confidence with which there is a difference. You can't always say there's absolutely this many on versus that many. Now, we could talk in those terms. We're talking more about compensation. We're talking more about, do you have the exact same floor on the surface of probably the exact same material, right? Otherwise, they will have slightly different fluorescence intensities under the same inspection. A polystyrene particle is not the same as a cell. But if we're looking at this and we say, this population is 30,000 times brighter than that population, and we have a 999 confidence on it, we can say that. And that's the beauty. That's the power. That's the whole reason why many of us are even in this field. Now, I've taken 2 different particle populations and I've gated them. And we're going to say, okay, it kind of looks like that 17237. Is that similar? 1354. Yes, they're on the same order of magnitude, right? So they are. And now we can say how much statistically similar this MEF value, or really this MEF value, is to this MEF value, because those are both the calculated ones that I'm running. And this one down here, that's more similar to like population 4, right? If I'm looking at population 4, I'm getting about 5,000, about 2,000. I'm on the same order of magnitude. Maybe that's more like population 3, between 3 and 4. Boom, we have something confident that we can say about our research. That's really the power of what we're talking about here. That's why we care. And then, like I said, to wrap up, we're talking about Q and B values, low-end sensitivity for your instrument. You need a second dim population, usually a second blank in your series. We can look at some formulas here. Usually this Q, sensitivity, B, background light levels. And we're talking about how well we can see down here, right? At what point is this all blended? At what point are we actually resolving meaningful information? And if we look at two dim populations, how confident can we be about the two of them? We can discuss this personally if you would like to, but I do find interesting, sometimes people will ask me, what's a good value? What's a poor value? And I always like to go experimentally with this. So if we just look at this Q quickly, CV. A CV for a dim population, a blank population might be 30%, might be higher. But like when we're looking at that first peak here, right? It's almost like a decade log. The CVs, where are we, right? The CVs might be 50%, something like this, maybe even larger, right? We're going to be squaring this number. So like 0.5 squared, you get like 0.25, usually about like 0.03, right? And you're multiplying that by the MEF value. Usually these second peak MEF values are 2 to 3 orders of magnitude. They're 50, 600. They might be 1,000 in a few channels. You're not going to be finding them much larger than that. So if we look at this formula and we are taking a small decimal times a 2 or 3 digit number and 1 divided by that number, we're going to be looking at a good Q value, as we see here, like 0.08, maybe even like 0.1 be a good value. What's a value that would be very, very poor? Well, if this CV was extremely large, but this MEF was also very large, as we saw, typically brighter populations have tighter CVs and dimmer populations have broader CVs. So if our Q value is something like 50, it's too good to be true. It means you have a very, very dim population that's extremely regular. And that's not something that flow cytometers usually measure. On the other hand, if you're getting a very, very, very small value, like 10 to the negative 4th, right? Something like that. Again, that might be too poor. You're not going to be able to resolve that. You're not going to be able to say anything statistically significant about that population. So we can kind of go back and forth about like what is a good one or a bad one. But as I said, longitudinal data is your strength here. It's always going to be something that you rely on. And I think that we are coming near kind of our end. I want to move towards questions or salutations because I want to leave at least close to 10 minutes. But I really appreciate your time. I hope this is a great resource for you. Again, thank you, Beckman Coulter, for hosting, and for Cassie.
Wonderful. Thank you, Michael, for your informative presentation. We will now start our live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box in the lower left corner and click Submit. We are close on time, so we will end up going a little bit past the hour, if that's okay with you, Michael, to answer some of these questions. Wonderful. Anybody that does not have time to watch this live, please note that this will be available to review on demand, so you can come back and watch what you might have missed. So I'll go ahead and ask our first question here. This question says, since ERF values depend on a flow cytometer's spectral filters, how is this accounted for when using the ERF beads mentioned?
Oh, this is really cool. I may have said that incorrectly. So the ERF, equivalent reference fluorochrome, NIST has actually chosen the fluorochromes that they care about, right? So they've chosen FITSI. I believe they chose maybe Nile Red or APC. You'll actually get a certificate that has, one of them is like a C30. So they're saying literally when they take our particle and they test it against this known quantitative dye in a fluorimeter, they're coming up with this ERF value. I do have some literature for this, but I think the question points to something that's very profound. It doesn't always apply to every channel, right? You might be able to say something robust about individual channels more robustly than you could potentially any other unit, or lean on their validation. But if they haven't tested something off of your laser or in your filter that you care about, like a PE Sci5, you could be out of luck. Now, again, my mentor did make a very cool and robust adjustable filter parameter template that is available on our website. Near the bottom of that template's homepage, there's one for the URQP. So you can literally type in, my filter is this and this, like it starts at 520 and ends at 575. And it works in much of the time, not all of the time, because there's only so much data that NIST provided to us, but it is more robust than what may appear narrow on the certificate.
Okay, excellent. Our next question here says, if we're measuring extremely small particles, less than 10 micrometers, and we're having a hard time distinguishing background noise from our populations, how would you recommend going about gating, or what products would you recommend for determining what background really looks like on our machine? We have a Cytoflex and generally use FITC to quantify virus-like particles.
Perfect. And I'm so glad you mentioned that you're using FITC. We have a tool called NFPPS, nanofluorescent particles. I'm very sorry. It's like a nanofluorescent size standard kit. It's an NFPPS, right? And it has particles that even go down to like 130 nanometers. We have an extra one off the end of it. It's like NFPPS-0152-2. But the kit comes with about quarter micron, half micron, 750 nanometers, and 1 micron, and it's FITC labeled. So if you run these beads on side scatter and FITC, you can pull out the populations by that fluorescence marker, and then you can see how far it is into that noise in your side scatter, and then you can gate them. So although it may appear sometimes when you're just looking at front scatter versus side scatter, it's all debris, it's opaque, you can't find it. Once you have that gate from the size standard saying there's these 3 or 4 gates in that low end sensitive range, you can then potentially say something more significant. And again, I could support you directly, but NFPPS, the nanofluorescent size standard kits.
Great, thank you. All right, our next question here says, what medium do you recommend for dilution? The about 2 drops in, 2 milliliters as presented earlier.
MilliQ, you could use your sheath fluid, but on a lot of these Coulter cytometers on the life science side, they're going to be using MilliQ DI water. You could use a little bit of, BSA or like sheath fluid, like I said, but usually I just am using DI water, MilliQ.
Great, thank you. All right. We have one more question here that we'll go ahead and ask before we wrap up. This question says, what are the most common issues encountered when aligning a flow cytometer, and how can they be mitigated to ensure consistent data quality?
Very interesting. What are the most common? It might be when you first run it, you don't see any peaks. You can't seem to find your population. Maybe your voltages are because these are quite bright beads, and if you're only used to running cells and you've kind of optimized and pushed your voltages up, maybe you don't see any peaks. Maybe you have a channel where it's completely blind. You may need to reduce your voltages all the way down and kind of stepwise them up until you can find that peak and set it where you need. They are typically quite robust, so usually it's not finding the population either through front scatter, side scatter. Side scatter is very sensitive. But thankfully, the particles are about 3 micron, which is usually near enough a cell size that you won't have too many problems.
Excellent, thank you. I want to thank you for your time today and your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and any submitted during the on-demand period will be addressed via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.